Welcome to the deep dive. Today we're diving into microRNA. You've been researching it, and getting ready for this has been, wow, pretty amazing.
Glad to be here. MicroRNA research is really interesting, always finding out new stuff about genes.
It's like discovering a secret language our cells use, all thanks to these little microRNAs. You shared some amazing research, Nobel Prize articles even, and I think by the end we'll see how microRNA is like a key to future medicine.
Definitely. We could target the root of diseases, make treatments incredibly precise.
Okay, so for everyone listening, molecules tinier than DNA, like secret agents controlling which genes make proteins, and proteins, well, they're vital for, pretty much everything.
That's microRNA, basically, like conductors, fine-tuning an orchestra of genes.
I like that. But before we go too far, let's start at the beginning. I always love hearing about these big discoveries.
Often, it's a simple question, something curious. For microRNA, it was strange worms.
This is fascinating. Scientists Victor Ambrose and Gary Rifkin, they were working with roundworms, C. elegans, really common in genetics. And they saw some were huge, others were tiny.
Imagine that under a microscope. A real head-scratcher.
I bet their notes were full of questions.
Totally. They checked everything but no answers. Then, they found a tiny RNA molecule, later called Lin4. It wasn't acting like any other RNA.
And that's the breakthrough.
Yes. Lin4 didn't make proteins, it controlled another gene, Lin14. Like a dimmer switch, it turned Lin14 down,
Yeah.
and that affected the worm size.
Totally new way of controlling genes.
Exactly. A huge shift, but, big discoveries. People were skeptical.
I can imagine. Tiny RNA controlling genes. Really?
Right. But then, they found Let7, another microRNA. And it wasn't just in worms, it was in lots of species, even us.
Yeah.
This wasn't just a worm thing, it was fundamental.
And that's Nobel Prize stuff. MicroRNAs changed how we see genes working. Thousands of them, each maybe controlling multiple genes.
Like, realizing life's instruction manual is way more complex than we thought, and we're just starting to read it.
So, small but mighty, these microRNAs. The secret influencers of our genes. But how do they work? Let's get technical.
It's a multi-step process, all happening inside the cell. We call it microRNA biogenesis, how microRNAs are made.
It all starts with DNA, right? Our genetic instruction manual.
Absolutely. Just like genes, the instructions for making microRNA are in our DNA. But here's where it changes. Instead of becoming proteins, these microRNA instructions go on a journey to become, well, microRNAs.
Okay, so we have our microRNA instructions encoded in the DNA. What happens next? So we've got these instructions for microRNA in the DNA. What happens then? It's like a tiny factory in our cells, right?
It really is. First, the microRNA DNA is copied into a long RNA molecule, a single strand, like a blueprint copy. We call this primRNA. Then it's trimmed and processed inside the nucleus, the cell's control center.
Okay, so shaping the microRNA, making it smaller, but it's stuck in the nucleus. How does it get out to work its magic?
Good question. This is where an enzyme called drosha comes in. It's like a pair of scissors. It cuts the primRNA into a shorter hairpin shape. We call that primRNA.
Drosha. Love the names. So the primRNA, our little hairpin, is it ready for action outside the nucleus?
Almost. It has to get out of the nucleus first into the cytoplasm. That's where it can regulate the genes. There's a protein, exportin 5. It's like the escort. It guides the primRNA through the nuclear pores out into the cytoplasm.
Wow, so many steps. It's incredible how it all works. But we're not done yet, right? It still has to become a real functional microRNA.
Exactly. Now, in the cytoplasm, another enzyme steps up, dicer. Dicer, just like drosha, is like a tailor making the final cut. It cuts the loop of the hairpin, and we get a mature, double-stranded microRNA.
Our star player is ready, the microRNA. Now what? How does it actually control the genes?
This is the exciting part. The microRNA teams up with proteins, forming a complex. We call it RIC, RNA-induced silencing complex.
RASC sounds kind of ominous. So this RASC complex, how does it silence genes? Not with a whisper, I bet.
Not a whisper, but you're thinking along the right lines. You see, each microRNA, it has a special sequence, like a fingerprint.
Right, that's how they find the right genes.
Exactly. Inside RASC, the microRNA is like a guide, taking the complex to messenger RNA molecules, mRNAs. Those carry instructions from the DNA to make proteins.
So the microRNA, with its sequence, finds the matching mRNA. Then what?
Once it's bound, the RASC complex can silence the gene in two ways. One is translational repression, like a roadblock. RASC stops the mRNA from being read, so no protein is made.
Instructions, but nobody to read them.
Right. The second way is, well, a bit more final. mRNA degradation. RASC doesn't just block the road, it demolishes it. It calls in enzymes to break down the mRNA so the protein is never made.
Wow, so they can either block or destroy the mRNA, these microRNAs. Such power in such a small package.
And it's even more amazing because which method they use, blocking or destroying, it depends on the specific microRNA and its target.
Seems like we're just starting to understand all of this, but knowing that they're so important for gene expression, what happens when things go wrong?
That's what researchers are working on now. Because microRNAs in disease, it's a complicated story.
What do you mean?
Well, they're involved in so many processes. If their activity changes, it has a domino effect. Sometimes microRNAs are like the good guys. Tumor suppressors, they stop cancer. But then some microRNAs are too active in cancer, making it worse.
So depending on the microRNA, it could prevent or cause cancer. That's wild.
It shows how complex it all is. And it's not just cancer. MicroRNAs are involved in heart disease, brain diseases, so much.
That makes sense since they affect so many things in the cell. Let's talk more about those diseases. I'm really interested in what you said about microRNAs and Alzheimer's, that kind of brain disease.
Alzheimer's is a tough one. Toxic proteins build up in the brain, and that causes problems with thinking and memory. New research suggests microRNAs might be involved in how these proteins are made and cleared away.
So if we could control those specific microRNAs, could we slow down Alzheimer's? Maybe even stop it?
It's a hot research area, and there are some promising leads. Scientists are trying to develop drugs that either act like good microRNAs or block the bad ones.
Like a whole new type of medicine based on microRNAs, tailored to a person's genes.
Exactly. And this isn't just an idea. Scientists are making progress. One way is to deliver synthetic microRNAs, or molecules that affect them, right to the specific cells.
Targeting the disease at the root, the genes themselves. Incredible.
That's the hope. But there's still challenges, of course. Getting these microRNA treatments to the right place at the right time and making sure they don't have side effects. It's tricky.
Like any powerful tool, we need to use it carefully.
Yes, but the potential is huge. Imagine treating cancer, Alzheimer's, even viruses in a whole new way, more effective and targeted.
This is so exciting to follow. So for everyone listening, we're really digging deep into microRNAs today, these tiny molecules with a huge impact.
We've looked at where they came from, how they work, their roles in keeping us healthy and making us sick, and their potential to change medicine.
And we see how they can be good or bad, shaping what our cells do and whether we get sick.
Plus, we've seen a glimpse of the future, where we might use them to create personalized treatments based on our unique genes.
Now, talking about unlocking secrets. There's something else fascinating about microRNAs, something we haven't even touched on, viruses. It's a microscopic battle. It's like a game of cat and mouse. These viruses, they've even figured out how to use microRNAs.
You got it. Some viruses, they're sneaky. They've evolved to use our own microRNAs against us. They can hijack them to help themselves copy and survive.
It's mind-blowing how they've been changing and adapting with us for, well, forever. So it's like a constant fight, right? At the smallest level.
That's a great way to put it.
Mm.
But we're not defenseless. Our immune system, it's got weapons too. And you guessed it, microRNAs are part of our defense.
We have microRNAs that attack viruses. Seriously.
It's true. Our cells, they can make specific microRNAs that target the virus's RNA, basically disarming them, stopping them from copying themselves.
That's incredible. Our own little defense system right inside our cells. This is amazing, this whole world of microRNAs. So, thinking big picture now, all this knowledge, where does it take us?
This is what gets me excited. We're on the edge of a whole new way of doing medicine, using microRNAs to create treatments that are super specific, personalized.
Okay, tell me more. What could that look like?
Imagine we could shut down the bad microRNAs, the ones that cause diseases like cancer. We could stop those signals that tell cells to go out of control. Or we could boost the good microRNAs, the ones that protect us.
It's like a whole new category of drugs designed around microRNAs made for each person's genes.
Exactly. And this isn't just a dream, it's happening. Scientists are working on delivering synthetic microRNAs, or molecules that change how microRNAs work, right to the specific cells that need them.
So we could treat the disease at its source, the genes.
That's the idea. It's not easy, though. Getting those microRNA treatments to the right place, making sure they don't cause more problems, it's a challenge. But we're working on it.
It's like anything powerful, we have to learn to use it the right way.
Exactly. But think of what we could achieve, treating things like cancer, Alzheimer's, even those viruses we talked about, in a completely new, effective way.
It's an amazing time to be learning about this. So for everyone listening, we really dove deep today. Who knew these little microRNAs were so important?
We started with the basics, how they were discovered, their journey inside the cell, how they can be good and bad for our health, and their incredible potential for medicine.
We saw how they can protect us and also cause problems, how they affect what our cells do and whether we get sick.
And we even got a peek into the future, a future where medicine is personalized, treatments designed for your unique genes.
It just shows how important science is, how asking questions and being curious can lead to breakthroughs that could change, well, everything. Who knew something so small could be so powerful?
And the best part? We're just getting started. Every new discovery leads to more questions, more to explore.
Exactly. The more we learn about microRNAs, who knows what other amazing things we'll discover about, well, about life itself.